my group is an exploratory virology group at NIBR, so we're working on emerging viruses, and one of the other aspects of the work that we do is pathogen discovery. And so you might have a question as why at NIBR are we working on pathogen discovery? Well, I think that from prior talks that people have given here before on pathogen discovery, as well as the talk I'm going to give today, we can all agree that identification of novel pathogen disease associations can really dramatically alter the landscape of opportunities for tackling diseases of unknown etiology. They allow us to develop new tools for potential insight into the mechanisms of pathology. If we know there's a viral agent involved, we can actually test for it and distinguish cases that involve that agent from those that don't. It also allows us to develop rapid assays for detection, monitoring, and control of disease transmission, as we saw with previous talks. And of course, targets for disease treatments, which is my organization's bread and butter, as well as potential routes to eradicate or prevent disease. So for our group, one of the big questions that we're thinking about pathogen discovery in is what's going to be the next agent that we want to be developing antivirals for? And as we think about that, we're trying to think about looking for agents in a very careful way. And where are we looking? Well, one of the places that we look is acute infectious diseases of unknown cause. And I'll talk today about outbreaks, primarily outbreaks in clusters of cases with hallmarks of infectious origin. And here is shown what I'll be talking today about an outbreak of a disease in horses that we found really interesting to follow up. But we're also interested in chronic diseases of unknown cause with biological precedence or plausibility for a case for an infectious agent being involved. And probably one of the most obvious examples of this is cancer. And in our group, as well as others looking at pathogen discovery, we all are kind of interested in how big is the slice of the cancer pie that may actually be infectious in origin. We know things like papillomaviruses and the hepatitis C virus have been involved in cancers. What other cancers out there may also have a viral cause? We're also thinking about other disease states, such as immune disorders or inflammatory states that have the hallmarks of an infectious state, but no known agent has been identified. So these are kind of the broad areas where we've been looking. And what I'm going to talk about today is sort of one of the very first studies that we did as our team was getting up to speed and building up at NIBR, looking at pathogen discovery to give you a sense of the types of work that we're doing here. So that disease area was Theiler's disease. So one of the many questions I've gotten since I've gotten here and people have seen me is, horses? Why are you looking at horses? At the time we started these studies, Novartis had an animal health division. And so our interest in diseases of unknown cause spanned from both humans into animals. Moreover, animals have been a really instructive model for us for many of our infectious diseases. And so this was an area that we thought we might be of some high interest for us. And this disease is actually really interesting. It was first described almost 100 years ago by Arnold Theiler. It's an acute serum hepatitis in horses that occurs primarily in outbreaks. And what happens is typically horses are administered some sort of serum or biologic product as a prophylactic treatment. And within 1 to 3 months after the treatment, they start to see cases of hepatitis coming up. And this has been thought to be viral, yet over the spectrum of almost 100 years since it's been first described, no agent had ever been identified when the opportunity to pursue this came up for us. Now, before I go into all the details of the project, I want to highlight the 3 key people that were involved in this study. So on the NIBR side, we had 2 investigators, Sanjay Chandriani, who's actually since moved on to BioMarin, and is doing more host-related biomarker development work, and Peter Skews-Cox. He's our informaticist. So Sanjay piloted all the work on the lab side, processing the specimens and pursuing the follow-up. And in conjunction with Peter, Peter carried out the analysis and fed back and forth with each other to really move this project forward. Now, this person here on the end is Christopher Bud Tennant. He's an amazing emeritus faculty member on the School of Veterinary Medicine and is actually one of the people who is a world expert in hepatitis. He founded the woodchuck model for studying, animal model for studying hepatitis. So he was our big link to the actual outbreak that happened that initiated this whole study. So what was that outbreak? Well, it was actually something really local. We had a horse farm in Nevada where there were a series of horses that over a short period, it was basically a farm where horses were boarded, and over a short period of time they noticed a series of symptoms arising in the horses that looked like botulinum toxin poisoning. And it turned out to be the case that with follow-up on this, they found there was a dead cat that had been exposed to botulinum toxin that was, had basically crawled into the hay that was their feed, that was the source of this problem. So on identifying this botulinum toxin poisoning in the horses, the veterinarian associated with this farm did what any good veterinarian would do and initiated administration of horse serum that contained antibodies to the botulinum toxin. This is a pretty common practice. It's a way to prophylactically treat the horses and prevent any other horses from suffering from the toxin. Now, what I want to point out here that's notable is that there are actually two symbols for the actual anti-serum treatment, and that's because the veterinarian started vaccinating or treating the horses with one batch of botulinum toxin serum, and that actually ran out. So then she switched to a second batch and finished treating all the horses. And this actually worked very well for them in terms of moving forward with preventing further poisonings. But within a few weeks, 2 horses came down with signs of hepatitis. And those 2 horses had gotten the second batch of antitoxin serum. Unfortunately, the one that had been administered to many of the horses. And then at that point is when she contacted Dr. Tennant in Columbia, in Cornell, who's the expert. And they worked together to begin to then test all the other horses that had received that second batch of antitoxin serum and found that almost 50% of the horses actually had signs of serum hepatitis. So this is a classic presentation of Theiler's disease. And as you can imagine, this isn't something that happens every day. And so Dr. Tennant jumped on this and worked very closely with the veterinarian to very closely monitor the horses in real time as the outbreak was occurring and unfolding, and linked up with us about following up to do pathogen discovery on this. So what we ended up doing early stages of the outbreak is getting an aliquot of the actual antitoxin that was suspected to be causing the outbreak, as well as samples from 2 of the horses that showed clinical signs of disease, jaundice, et cetera. We had aliquots of that serum, and at the time we had no idea, of course, what type of virus might be present. So we extracted the aliquots with phenol, triazole, as well as a virus-specific sort of preparation method just to cover all our bases. We generated RNA and DNA from all of these and used massively parallel sequence analysis to try to identify if there were an agent present. And basically, we took the RNA and DNA, the total sample that was available from all the preps that we had done, and did random amplification with primers that were tailed with the Illumina-specific primers that are required for generating libraries and doing deep sequencing on the Illumina sequencing platform. And so here what we're doing is we're just basically getting a metagenomic sample of what's present in the plasma of these horses at the time that these were sampled. And just for those in the audience who are a little less familiar with this type of approach, when we talk about metagenomic sequencing analysis, we're talking about sequencing all the nucleic acid that could be present. Now, in these samples, it could be material from the horse, bacteria that might be present, virus, hopefully, and then there may be unknown sequences that have never been seen before, and that's why we have no idea what they are, as well as some indistinguishable sequences, such as repeat sequences or other sequences that might be of low quality. So this is what we expect to be present given the randomly primed amplification approach that we choose, but it's not going to be that easy to identify because when we actually do, these samples might be, these different species might be present in different amounts, and this might be very small relative to the amount of horse or bacteria that might be present. And likewise, all the samples are in very short fragments, as is required for the library prep. So a big challenge for us is to be able to identify the needles of potential viral sequence in this haystack of all the other sequences that are present in the samples. And so how do we do that? Well, as many people in this field do, one thing we do is there's a lot of information that's known in the databases, so we can remove all the sequences that we know about and then take what's left and interrogate it for the presence of viral sequence. And so what Peter did was he basically went to NCBI and found a draft genome for a draft sequence for a horse genome and used that to deplete the known non-viral sequences. And he used BLAST to do that. So he just took, this is very highly related to a horse. You can do a nucleotide BLAST, identify all the reads that correspond to horse, and just filter those. So that's the first step. And then the next step he took was to basically develop some code to deplete problem sequences. So as I mentioned, these sequences that are going to be indistinguishable. So things that either are low quality with ends, things with low complexity repeats, or just straight up repeat sequences. Those are just going to be junk that are going to align to everything and be confusing. So he basically developed some complexity filters to get rid of those sequences. And so then the next task for us is to be able to, kind of move forward and take this much smaller group of sequences and look for anything that might be known, look like or similar to a known virus that we know about. And so again, his strategies for doing that are just simple BLAST analysis. So the first level analysis is there anything in there that looks like a known virus that is in existence that's in the database? And so he just took all the reads that were left over and then blasted it against a database of all known viral sequences. And what we're showing here is just, of course I have a viral cold right now, so what we're showing right here is now just sort of an example. Like if you have something that's, even if it's a short read of a couple hundred bases, if you have something that's highly similar to something that's already known, it's going to be very easy to detect. And what he found was, unfortunately, no viral signature in any of the samples when he's looking at the nucleotide level. Well, that's pretty stringent, right? You're only going to be able to see things that are getting, when you get to the level of 75 to 80%, you start to lose confidence if you see something that matches by BLAST-N. Your actual probability that's a real match is lower, and also your confidence is lower. So another trick that we can do to extend our analysis for things that are more diverse is translate all the sequences and blast them against the protein sequence. And so that's the next step. He then took all the leftover reads, translated them, and aligned them to all the known translated viral protein sequences. And here you can pick up things that are only very low percentage identity, but they're still matches. And when he did that, what he found was we actually did start to see some matches. And there are actually matches across all the samples that we looked at in the RNA fraction. So Mikey and Quesadilla are those two cases of horses that suffered acute clinical signs of jaundice and severe serum hepatitis. And then this is what we call the inoculum is actually an aliquot from that antitoxin 2 that was used that actually led to the outbreak. And so what you can see is we see a couple different viral families that are coming up with hits. So for Mikey, we saw almost 3,000 hits in the Flaviviridae family. The Baculoviridae family, we likely think is actually related to contamination from sequences that were derived from the enzymes in the kits that we use to do the library preps. Similarly, Quesadilla, we found on the order of 3,000 hits to Flaviviridae, and likewise the inoculum, we also saw hits. So these corresponded to a small fraction of the approximately 22 million reads that we got from each of the samples. So on the order of 1,000th of what was present in our initial data set that we could actually recognize as maybe a viral sequence. And so what do we do with that? Well, first, before I go into that, Flaviviridae. For those of you who aren't familiar, this is a viral family that corresponds to envelope viruses that are primarily spread through arthropod vectors, or mosquitoes, or ticks. They're monopartate, single-stranded positive sense genomes, typically 9 to 12 kb in length. And some examples that are probably familiar to people here at UC Berkeley are dengue virus, but yellow fever virus, West Nile virus, and also hepatitis C virus are kind of key members of this family that are pathogenic. And it didn't escape our attention that hepatitis C virus happens to be a hepatitis virus that's in this family. So Peter took those reads and basically moved them into an assembly pipeline using an algorithm called Price that was developed at UCSF to basically take the reads that he had identified and then use those to seed assembly of contigs against the whole set of viral reads that were obtained for each sample. And by that way, he would be able to pull out reads that maybe we didn't identify because they were so divergent from what was previously known before, but they fit into our contigs. And so what he was able to do was pull that out, and in the end, for all 3 of the samples, assemble near complete genome coverage. So actually, Mikey was a sample for which we did get complete 10.5 kb genome sequence coverage. And then Quesadilla and the inoculum, we saw multiple contigs that didn't quite join up, but did have near complete genome sequence coverage. And so what I'm showing right here is a schematic of a typical filovirus genome where you've got structural genes here at the 5' end in the non-coding region, followed by non-structural genes that are required for replication. And when he took the sequences that he recovered from each of these samples, these showed 99% sequence identity across the entire sample, consistent with the idea that this is an outbreak. They all have the same isolate that was derived from an outbreak. The other thing Peter did was after he assembled a genome, he then took all the reads from each of the samples to assess how many reads were actually present. And what he was found was that there are order or 2 magnitude more reads present than he had originally identified without having the sequence. These are highly divergent agents. So one thing that was happening in parallel with this, literally like the day he was assembling these genomes, we found this paper that came out about novel Hepatitis, which are also flaviviridae viruses that had been identified by a separate group. And what you can't really read here is that a number, so this group had previously identified a flavivirus in dogs, developed a serology assay, and then tested a whole spectrum of different types of animals. And of those animals, they found serology positives in horses. And among those serology positive, many of them, they were able to fully recover genomes from horses. And so one of the first things he did when this sequence came available was look to see how similar our virus was with those horse viruses. And so our virus, which we would call Theiler's disease-associated virus, is over here on this filovirus phylogeny. Whereas the viruses identified in this other study that were called non-primate hepativiruses were identified here. And this here highlights where HCV maps on this. It's interesting that our virus maps closer to these viruses called the GB viruses. These are a group of viruses called PEGI viruses for persistent viruses that were originally identified in early days when people were looking for the causative agent of HCV, but have never been associated with a disease. And it's interesting that these viruses were found by sort of just serological survey of viral serum, not viral, horse serum that was available randomly through general testing of horses and unlinked to a disease. So it's an interesting flip where the disease-associated virus is actually found among those that have previously not been identified as disease viruses, and vice versa for the other horse virus. So that's where we were at that point. And so we have these 2 different viruses, and primarily we were mostly concerned with our virus and wondering, okay, is this really a pathogen? And so there are a couple different things that we typically do in the studies that we're looking at, because we're always interested in identifying specific virus-disease associations, to try to actually link that as whether or not it's a pathogen. So one of the first things we strive to do is just confirm that the novel agent that we might identify is actually involved in the disease. And that involves, first of all, physically recovering the virus or virus sequences from specimens where it was originally identified computationally. We always want to make sure that what we're seeing and hypothesizing as a novel virus isn't actually 2 different viruses that we've never seen before, or that there's no errors in our computational prediction. The next thing we want to do is also detect virus, ask whether or not we can detect virus-disease association among the initial as well as independently collected specimens to solidify the association. And finally, if it's feasible, obtain biological or immunologic evidence that the virus infection is consistent with disease that we're looking at. So Peter and Sanjay worked really closely together to design different kinds of PCR primers to be able to PCR overlapping fragments from our clinical specimens to validate this sequence. They were successful in isolating these overlapping contigs and sequenced those by Sanger sequencing, and those agreed completely with what we found from the computational or predicted sequences. So we at least validated that first part. We then went on to develop a series of robust qRT-PCR primers to actively screen across specimens in our initial outbreak set, as well as independent specimens, to look at the virus-disease association. And those data are shown here. So these are the results of a qPCR assay where, just to orient you, the y-axis is inverted so that the low numbers are at the upper end, which signify high viral loads because you have a low CT value. And the high numbers for the CT raw data are at the lower end of the axis, indicating low viral load. So one of the first things we did was look at farm A, which is the outbreak farm. And on farm A, what we found is when we did the qPCR assay, all the samples from horses that had received that antitoxin 2 were positive with very high viral loads for having Tdap present. All the samples all the samples derived from horses that either received the first antitoxin or were untreated had very low CT values and virtually no virus. Now you'll note that there's 2 samples here that received that antitoxin 2. Those samples actually were positive with one, but not both of the assays, so they're sort of borderline, and we kept them in the so they map to this low viral load section. We also looked at a random distinct farm to see how prevalent this virus was. Is it something that's present in all horses when we randomly sample serum? And what we found there was that very few of, none of the horse serum that we identified in that set of 20 horses were positive for Tdap. So it does seem to be very specific to the antitoxin and to this farm outbreak. What was great about Bud Tennant was he not only knew the vet at the outbreak, but he also knew the supplier of the antitoxin. So he was able to actually go to the antitoxin supplier and identify the specific horses that the serum was generated from and give us those samples to test. And there are actually, so this is actually the antitoxin 2, the original aliquot. And there were 3 horses that generated the antitoxin. As you can see, there's one horse that has very high viral load. That's probably the source of the contamination of that antitoxin. And of course, water has no virus. Now, the other aspect of linking this to the disease is what fraction of these horses that have actually been delivered to have had antitoxin 2 and show virus show signs of hepatitis. And that's shown here. Pretty much, as I mentioned, half of the horses showed signs of hepatitis. So you can see a number of horses here. The red stands for symptomatic clinical signs, and the blue highlights those that have subclinical signs. So of those horses that are symptomatic, many of them, all of them have high titers. This is one of those borderline samples. But interestingly, there's a whole set of horses that seem to be subclinical or asymptomatic. So while we have a good association between the virus and infection and disease, it's not 100% prevalent for all the horses. And this is not unusual. It's something that we've seen before with HCV. Those who contract HCV initially are able to clear it, and not everyone goes on to display symptoms. But aside from that, we wanted to gain more information into what the virus infection was doing. And our collaborators are actually able to obtain 4 ponies and do an inoculation study. So they took these 4 ponies, did some preclinical studies, then took the antitoxin botulinum serum and injected that into the horses, and then followed them for 10 weeks. And the question was whether or not they see clinical signs among any of the horses, if they could see any subclinical signs in the liver and in their blood, and then also do liver biopsy if they saw any clinical signs. And this is just a quick summary here. What you can see here with regard to the Tdab is there was quite a diversity of types of courses of disease with these animals. So some animals, like this one in green, 118, showed really low to no replication of virus, while others had slow replication and others had rapid remaining high levels. Some went down. So there's some diversity of the infection kinetics. And you can see that on the whole, the 3 out of the 4 horses did not go on to develop symptoms, not unlike what we saw in the actual outbreak. But we did see one horse that did go on to develop altered liver enzyme levels in the blood, and all those levels were preceded by a rise and high attained viral load. We also found that in terms of looking at the samples from a year out from the outbreak, that if you took horses that had low viral titers, if you looked a year later, they maintained low viral titers, suggesting that there wasn't horizontal transfer across the horses. And you can see here that many of the horses that had high viral titers actually went on to clear the virus. And then there's a small fraction, about 25%, that maintain viral load asymptomatically. So overall, in terms of the pathogen discovery study, we identified a highly divergent member of a flavivirus family in an outbreak of Theiler's disease. This was associated with a spectrum of signs and symptoms. About 50% of the animals showed signs of hepatitis. A small proportion had severe fatal outcomes. And about 25% of the animals went on to become chronic carriers. Again, very reminiscent of what we've seen previously with diseases like HCV. Our experimental inoculation studies with Tdab indicates variable kinetics of infection and suggests that there may be host determinants or some other factors that we don't understand that play a role. So in terms of like where this study was done, actually a couple years ago, and since then our collaborators have moved on to do additional studies, and there are a lot of remaining questions. So I think one big question that still remains is whether or not we can say that Tdab really is a causal agent in Theiler's disease. Here, we haven't done a formal demonstration of Koch's postulates in that we haven't purified the virus, developed an infectious clone, and reinfected a horse. And that really remains a gold standard. Right now, we can't rule out the possibility that there are other agents that may be present in the serum that we haven't detected or didn't have enough sequence similarity to what was known for us to pick up that could also be present. And the other question we have is whether or not Tdab is the causative agent or one of many causative agents. We see here in this outbreak that Tdab seems to be playing a role, but what about other outbreaks or other types of viral diseases associated with serum? So as I mentioned, there's MPHV. I didn't mention this in the talk, but we did test all our samples, and none of them tested positive for, well, only 1 or 2 from the unrelated farm tested positive for NPHV. So NPHV was not involved in this outbreak, but could it be involved in others? Likewise, there's an equine Peggy virus that was discovered after our paper came out and these studies were done. Does that play a role? And is there something going on similar to what's been described by Charles Chu at UCSF with human Peggy virus 2, a new Peggy virus that seems to be present in co-infection with HCV and may be associated with distinct outcomes with HCV. Could that be going on here with a separate virus that we've yet to discover? We don't know. And there's a whole host of questions about the natural history of Tdab that still remain to be addressed. Is Theiler's disease the whole story? Are there sporadic cases of liver disease that Tdab may be causing? And what happens to chronic carriers, and what are the factors on the host side that may influence disease progression? What's exciting for me is that although we didn't end up following up on this because our animal health division was sold, our clinical collaborators and actually a local Bay Area investigator who originated from the Delaware lab have moved on to extend these studies and look at the epidemiology, additional viruses involved in horse serum hepatitis and other diseases, and have recently been awarded a grant to kind of really thresh out some of these questions on the natural history side so we can get a better sense of what's causal across many different types of these diseases that we're seeing. With that, I'll acknowledge the external collaborators and everyone involved in the study and take any questions.
Hi, very nice talk, thanks. 2 questions. The first, how do you think this virus is transmitted? Is it like dengue and yellow fever through mosquitoes or not through HCV? Second question, what is the structure of the UTRs, the 5' and 3'? Does it have a long 5' like HCV containing an iris or a short one? Thank you.
Yeah, so the first question was where does the virus come from?
How do you think it's transmitted?
Oh, the first question was how do you think it's transmitted? Yeah, I was going to put that on there as one of the questions because we don't know. We don't know about the natural history. Where did it come from? Right now, what we know is that it was in antitoxin, antiserum. Our collaborators have looked at other antitoxin, antisera, and they have found it in other farms that are generating the antitoxin serum, but the actual source of that is not known. And the fact that we're not seeing horizontal transmission, they've done some other separate studies. They're not seeing it in the context of any sort of exposures outside antitoxin exposure at this point. So it's not clear where the origin of the virus is from. And in terms of the 5' UTR, the 5' UTRs are somewhat similar to what we've seen for flaviviruses, but I don't remember all the details. And we weren't able to get the 5' UTRs on all of the isolates, although presumably they're the same isolate, so they should be the same.
I guess. So I guess it's a dovetails on the previous question, but the lack of horizontal transfer, does that imply that this is probably vectored by another organism? And also, have you analyzed the 3' UTR of this flavivirus to see if it groups with the flavies that seem to be transmitted or go between mammals and insects, or one that groups more with just mammals or more with just insects?
Okay, so I can answer the latter question. We haven't done a detailed analysis of the 3' UTR, so I can't tell you whether it segregates with that regard to the different types of vectored flaviviruses. And then the first question was about the horizontal transmission, and does that?
Just the lack of it, does it imply that it's vectored?
I'm not sure it implies that it's vectored or not. It doesn't rule it out, but it doesn't imply a simple, basically what it implies is no simple horse-to-horse transmission.
Hi, I'm just curious if you've actually done any follow-up work to see if this pathogen might actually appear in other species besides horses.
We have not done a lot of follow-up on this. Our collaborators have primarily looked at horses, so we don't know about the prevalence in other species, as far as I know. Good question.
Hi. I was interested to see to hear how the chronic carriers are doing. Have you had a chance to follow them, and do they eventually develop disease, or are they completely asymptomatic?
We followed them out to 1 year, and they were asymptomatic or subclinical, and there was no major change in their disease state. We haven't. I don't know what their status is now, though.
And have you looked at any immune correlates, like between the clearers and the non-clearers, to see whether there's anything we could learn about HCV clearance and non-clearance?
Sorry, I couldn't hear it.
Immune correlates, the immune system, to see how they, yeah.
No, that we have not looked at that yet. I think that's sort of the downstream work that's going on now.